Welcome to Dewdear Academy. I am discussing a Kerala PSC exam question, the DHS exam question paper, one of the question I have selected, and this video will be useful for all people who are preparing for different competitive nursing examinations, especially in nursing. So the question is, modified acid fast staining commonly used for staining Mycobacterium tuberculosis is. Options are Seale-Nielsen staining, Gram staining, negative staining, and the option D is none of the above. The question is, modified acid fast staining which is commonly used for staining Mycobacterium tuberculosis. So here the correct answer is option A, that is Seale-Nielsen staining. And we are going to discuss each of the options and also the correct answer in detail. So the first option given here is Seale-Nielsen staining. What is Seale-Nielsen staining? It is a type of acid fast staining. It is a type of acid fast staining, and it is mainly used to identify Mycobacterium tuberculosis. Mainly it is used to identify Mycobacterium tuberculosis, and also the other Mycobacteria can be identified using this Seale-Nielsen staining. So what is the speciality of this bacteria? Mycobacterium tuberculosis means they are having waxy lipid rich cell wall. You can see this Mycobacterium tuberculosis means they are having lipid rich cell wall. So what happens is that is making resistant to other typical staining techniques. The cell wall of this Mycobacterium tuberculosis means this mycobacteria has a lipid rich cell wall. So that is making resistant to getting stained with other common staining techniques. So this type of acid fast staining is usually effective for Mycobacterium tuberculosis, mainly this Seale-Nielsen staining. And what is the primary stain used in Seale-Nielsen stain means the carbol fusion is the stain, the primary stain that is being used in Seale-Nielsen staining. So this type of stain means it can bind strongly to the cell wall of Mycobacteria. This carbol fusion stain can bind strongly to the cell wall. And after staining, the slide is washed with an acid alcohol solution. So it will be decolorized. The non-acid fast cells are getting decolorized if it is washed with acid alcohol solution. And what happens? The only acid fast bacteria will be stained red. So you can see this in the picture. First, when we are adding the primary stains, so this is the procedure for the Seale-Nielsen staining which is used for the Mycobacterium tuberculosis and all. So when we are applying the primary stain, that is carbol fusion, we can see the picture like this. This is how it will be happening, and after heating using the flame, after heating, and then we are adding the alcohol solution. We are adding acid alcohol solution to decolorize. So what happens means it will be the non-acid fast cells are getting decolorized. The non-acid fast cells are getting decolorized, and the acid fast bacteria get stained red. All the acid fast bacteria are getting stained red. So if we are again applying the counter staining also with the methylene blue, we can see the results like acid fast will be becoming the same color. Stained, it will be stained red, and the other become blue. So this is the procedure for Seale-Nielsen staining. So this method is a key diagnostic technique for tuberculosis and also in other diseases which are caused by Mycobacteria. So what happens is the stain which is used, the primary stain is the carbol fusion stain, and when we are applying this stain, if there is any non-acid fast cells, they will be becoming decolorized when we are adding the acid alcohol solution, and the acid fast cells will be remaining red. So that is what we are seeing in this procedure, Seale-Nielsen staining. So next I am discussing the Gram staining. In the Gram staining, it is a common staining method. Gram staining is a common staining method. Mainly we are using this staining method to differentiate the bacteria into two, that is gram positive bacteria and gram negative bacteria based on the structure of their cell walls. We are dividing it into gram positive and gram negative bacteria using the gram staining technique. So what happens is, in gram staining, the gram positive bacteria retain a crystal violet iodine complex and it becomes purple colored. The gram positive is purple colored, while gram negative are decolorized, and when we are counter staining it, it becomes pink. So this is that procedure which we are using in gram staining. But what happens, the Mycobacterium tuberculosis, they do not respond well to the gram staining, because we know the cell wall is lipid rich cell wall. So it does not fit well with this gram positive, gram negative categories. So for the Mycobacterium tuberculosis, we are preferring the acid fast staining, mainly the Seal-Nielsen staining. The gram staining we are just using for the gram positive and gram negative bacteria to divide into gram positive and gram negative bacteria. So this is the gram staining. We can see the gram staining, the different steps here. So first, when we are adding the primary stain, the added to the specimen smear, what happens? The gram positive will be purple and gram negative is also purple colored. When we are adding the crystal violet iodine complex, when we are means if we are staining or we are adding this crystal violet iodine complex, what happens is, again the gram positive and gram negative appears purple. When the decolorizer alcohol is being added, what happens? The gram positive still remain purple and gram negative is colorless. The gram positive is purple and the gram negative become colorless. Then the counter staining using safranin. Safranin is the counter stain used here. On counter staining, what happens here is, the gram positive will be purple and gram negative becomes red or pink. The gram positive is purple and gram negative is becoming red color or pink colored. So that is what I have explained here. The gram positive is becoming purple colored and gram negative become pink or red. So anyway, this type of staining is not used for Mycobacterium tuberculosis. Then we have the negative staining. That is a technique in which the background is stained. In negative staining, what we are doing is the background is getting stained and the cells are unstained or they will be lightly stained. So the visualization of the cell morphology, size and arrangement is easily visible, as the background is only getting stained and the cells are not stained here. So this is the picture of the negative staining. The negative staining, we can see only the background. The background is becoming dark here, and all the cells are colorless bacteria. They are lightly stained or they are unstained. So we can easily see the morphology, size and also arrangement of the cells with this negative staining. So it is commonly used for observing the bacterial capsules. We are mainly using this negative staining to observe the bacterial capsules, not for staining the Mycobacterium tuberculosis or any acid fast bacteria. So then in negative staining, we are using the dye that is nigrosin. We are using the dye nigrosin or India ink. The dye that is used is nigrosin or India ink. So that these are the differences of different types of staining. Okay, so Seal-Nielsen staining is type of acid fast staining, and that is used for Mycobacterium tuberculosis because the Mycobacterium tuberculosis has a lipid rich cell wall, and this type of staining is only the stain using the carbol fusion will be strongly getting binded to the Mycobacterium cell wall, and it gives the different color changes. The gram staining is mainly used to differentiate into gram positive and gram negative. The gram positive means it will remain as a purple color ultimately. In gram negative becomes pink in color, and the negative staining means we are staining only the background and the cells are not stained there. So it helps to determine the morphology of the cells and also mainly to determine the capsules. And the negative staining, the stain we are using there is India ink. And the last option is none of the above. That is not the correct answer, because anyway Seal-Nielsen is the staining is the correct answer here. So I hope this video is useful for all of you who have been listening here, and you have got a different ideas regarding the different options mentioned here. So thank you all of you.